Okay, so for the H1 students, this is the last recording, right, for the enzyme lecture. Okay, we'll be looking at the metabolic pathways. Metabolic pathway is a series of reactions, each catalyzed by a different enzyme. Right, so in the example on the screen, it is a pathway that is made up of 3 enzymes. The first enzyme catalyzes the formation of intermediate A from the substrate. Then the product of enzyme 1, intermediate A, becomes the substrate for enzyme 2, and so on, right, until the end product is formed at the end of the metabolic pathway. In many of these metabolic pathways, feedback inhibition occurs when the end product accumulates. Okay, so when there is enough of the end product, it can stop the function of the metabolic pathway to prevent too much of the end product from being synthesized. Right, so in my example here, the end product inhibits enzyme 1. Right, so in other pathways, right, it can inhibit other enzymes. It doesn't always have to be the first enzyme. Right? In other pathways, sometimes it will inhibit an enzyme in the middle, for example. Okay, so this feedback inhibition, right, have advantages. It helps to prevent wastage of resources. When we have enough end product, then we don't have to waste the substrate, right, and convert it to even more end product, which is no longer useful, right? We already have sufficient. And also during the enzymatic reactions, there might be energy involved, right? So that would be a wastage of energy as well. Some of the intermediates, right, can be channeled to other pathways, okay, by inhibiting enzymes like in the middle of the pathway. Okay, so you have this picture in your notes, right? When there is enough product D, okay, so it can inhibit enzyme 2. Okay, so we still have enzyme 1 functioning, right? Substrate is still formed into intermediate B, but intermediate B is not catalyzed by enzyme 2 to form intermediate C due to the inhibition by product D. Okay, so it can be channeled to another pathway, right, for enzyme 4 to convert B to E and then to form product F. Okay, so this helps to channel the intermediates to other pathways to form other useful end products when one end product is already sufficient. And of course, sometimes too much of a good thing is bad, right? So if there is no such inhibition and too much of the product is formed, there may be potential adverse effects. Okay, so this is a metabolic pathway, and little bit on the feedback inhibition. I understand that in the H1 syllabus there's no inhibitors or inhibition, but sometimes questions in the A levels do ask beyond the H1 syllabus. So this is just an example to show you a bit of inhibitors and inhibition, right? So if it comes, right, and you would be better able to understand, right, the information or the case study given. All right, and it is a hallmark or a very common phenomenon we see in metabolic pathways anyway, right? This feedback inhibition. Right, so a question here, right, in your notes, right? Why do we have metabolic pathways even, right? Why must the reaction, right, from substrates to products be done in a series of steps? Why not just one step? Why do we need, for example, in this example, right, why do we need 3 enzymes to form the product? Why not just one enzyme to catalyze the substrate into the product? Okay, why multi-step? Okay, so there are some advantages to having a multi-step metabolic pathways. Firstly, again, right, a very common phenomenon in such pathway is the feedback inhibition. So the end product can inhibit one of the enzymes in the pathway, and that helps to, again, prevent wastages, allow intermediates to be channeled to other pathways, and also prevent potential adverse effects, right, of too much end products. And then also, right, when we have multi-steps pathways, it allows for a greater level of regulation or a more a finer control of the reactions. Okay, so if it is just one step, right, involving just one enzyme, then really all we can control is either we have the reaction or we don't. But when we have multi-steps, then we can have different points at which the pathways can be controlled, because each enzyme offers a point of control. We can have maybe reactions to occur. Maybe the first few steps can occur, but then not the last few steps, for example. Or maybe we can slow down the first step, increase the third step, then slow down the fourth step, for example. Right? So there can be more varied control rather than just on and off, if it is just a one step, one enzyme kind of reaction. Right? And as mentioned before, right, intermediates can be channeled to other pathways to form other useful products. And if the reaction involves release of energy, one example is respiration, okay, that having more steps and each step releasing a smaller amount of energy would allows for a better utilization, a better conversion of the energy into ATP for storage, and also to prevent damage to the cell. Right? So, for example, for respiration, if all the energy stored in glucose is released at one go in one step, then that increase in energy would result in a large increase in temperature inside the cell. The cell will burn. And of course, the cell would not be able to absorb the energy or make use of the energy very efficiently, because there's too much energy being released all at one go. But when the energy is released slowly, step by step, which is what occurs in the respiration pathway, which involves many steps, alright, it allows the cell to slowly absorb the energy, slowly make use of the energy, slowly convert the energy into ATP, for example, and also prevent a large increase in temperature that would damage the cells. Right? So having a pathway is essential in this case when there is a release of energy. Alright, so these are some advantages of having these multi-step pathways rather than a one-step reaction. Alright, okay, so to end off the lecture with the appendix, right, of enzyme cofactors. It's not really in the syllabus, but since we are looking at how enzymes carry out its function and knowing that most enzymes do require the presence of a cofactor, although this is something that we should know, don't have to memorize by heart. Okay, but if you do see cofactors in the question, then you understand what it is for. Alright, so there are different types of cofactors. Some of them are inorganic, and these are usually ions, but most of the cofactors are actually organic, right? And some of them bind tightly or permanently even to the enzyme, and these are known as the prosthetic groups. Some are quite loosely associated. They only come in to bind to the enzyme during the reaction, and they would dissociate once the reaction is completed, and these are known as the coenzymes. Alright, so these cofactors do play an important role to allow for the enzymes to carry out its function. Some of these cofactors are found near or at the active site, so they kind of complete the shape of the active site sometimes. Some are involved in the reaction. They help to facilitate some of the reactions that occurs. So they do play an important role in the enzymatic reaction. So I thought this is something that you should know. And also, right, next year when you learn about respiration, you will also come across some coenzymes. Right? So heads up here. Alright. So that's the end of the lecture, right, for H1 students.